Protein digestion. So let's first understand what proteins are made of. Over in the bottom right here is a protein known as hemoglobin. There's a primary structure, a secondary structure, tertiary and quaternary structure that make up proteins. I should say primary, secondary, and tertiary for all proteins, and some include a quaternary structure. The primary structure for proteins is the amino acid sequence, which we see up here. And that's going to be relevant because that needs to be broken down before absorption occurs within the small intestines. To be clear, proteins are polymers, which we see right here, a bunch of linked monomers. The monomeric subunit for proteins are amino acids. There are 20 amino acids in the body, which we see right here, some nonpolar, some polar, charged and not charged. If we take a protein, we break it down, the first thing we would do would be to unfold it, or the first thing the body would do would be to unfold it, creating what's known as a polypeptide chain, which is the amino acid sequence. And then enzymes will break that down into tripeptides and dipeptides, which are merely a chain of 3 amino acids or 2 amino acids respectively. And then those can be further broken down into the monomeric amino acid subunits. There are a number of different enzymes that break proteins down. There are endopeptidases that are also known as proteases that cleave the proteins in the middle, whereas exopeptidases cleave enzymes at the very end, as we see here with the aminopeptidase and the carboxypeptidase. Of the endopeptidases, there are pepsin, trypsin, and chymotrypsin as examples. Pepsin is secreted by the stomach, initially from the precursor proenzyme, also known as a zymogen. A zymogen is a proenzyme. The zymogen for pepsin is pepsinogen. It's the inactive form. And then trypsin and chymotrypsin are the endopeptidases released from the pancreas. As far as absorption goes, we can absorb di or tripeptides or free amino acids. The free amino acids, which we see in the middle right here, tend to be absorbed via sodium transport, utilizing the kinetic energy of sodium moving into the cell, and also a sodium cotransporter to move out the basal surface into the bloodstream. The di and tripeptides move in via a hydrogen cotransporter using the kinetic energy of hydrogen to move into the cell. It can also use a hydrogen cotransporter to move out of the basal surface of the cell, or sometimes these dipeptides or tripeptides further broken down by enzymes into their constituent amino acids. A third way of absorbing proteins is some of the smaller polypeptide chains, so this would be more than 3 amino acids, enter the bloodstream, move across the enterocytes via transcytosis, and that's merely the endocytosis of the protein into the cell and exocytosis on the basal surface directly into the blood.